Hello and welcome everyone. As we all by now have got a clear picture of the novel coronavirus or the SARS-CoV-2 in our minds, I'm here to talk about the pathogenesis and sail you through the journey of the virus inside the human body. The droplets carrying the virus may land into the nose or the mouth of an individual where it infects the nasal or oral mucosa by binding to the ACE2 receptors present there. According to a study by the Lancet dated 3rd August, the virus was found to infect the olfactory epithelium and enter the CNS via an olfactory bulb-mediated neuronal retrograde route and infect the olfactory gyrus in the brain, which was found to be the first area to be infected in the entire CNS. This can justify the olfactory dysfunction or anosmia as an early manifestation of COVID-19. The virus then travels down the throat and the airways, infecting them on its way, thus producing dry cough in the patient. When the virus reaches the alveoli, it binds to the ACE2 receptors present on the type 2 pneumocytes lining the alveoli with the help of spike protein or S protein. The protease enzyme named TMPRSS2 receptor or TMPRSS2 receptor is also present on the host cell membrane, which cleaves the S protein and aids in the fusion of the viral envelope with the host cell membrane. This fusion is essential for the entry of the virus in the host cell, which happens via endocytosis. The virus then enters the cell, the host cell, along with the ACE2 receptor and undergoes disintegration to release the viral single-stranded RNA or ssRNA. The viral RNA then undergoes polymerization with the help of RNA-dependent RNA polymerase to give multiple copies of the original viral RNA strand. The viral RNA also undergoes translation to give viral proteins. The viral proteins and the viral nucleic acid assemble together to give viral to form new daughter viruses. The newly formed viral particles are ejected out of the host cell with the help of viroporins. Viroporins are small membrane-embedded proteins that have ion-conducting or IC properties. What they do is they aid in viral replication, maturation, and also release. There are three types of viroporins basically, namely protein E, ORF3A, and ORF8A. Protein E and ORF3A being more dominant. The viroporins have IC properties and the ORF3A is therefore a potassium ion channel, and protein E itself forms calcium ion channels in the Golgi membrane or the ERGIC of the host cell, thus disturbing the normal calcium homeostasis in the intracellular environment. This stimulates the cytosolic innate immune signaling receptor known as NLRP3 inflammasome. NLRP3 inflammasome is responsible for inflammation and cell death known as pyroptosis. What exactly happens is that the NLRP3 inflammasome converts pro-caspase-1 to caspase-1, which in turn converts pro-inflammatory cytokines into their active form. Example, conversion of pro-IL-1 beta to IL-1 beta and pro-IL-18 to IL-18. This triggers a cascade of other downstream inflammatory mediators like IL-6, TNF-alpha, the prostaglandins and leukotrienes, which are all responsible for causing fever, and which is a key feature of COVID-19. There is also production of pore-forming proteins called gas dermins in the host cell. The gas dermins, along with the viral proteins like the ORF3A and protein E, contribute to the formation of pores in the host cell membrane. Through these pores, the cytokines are also released and the newly formed viruses are ejected out. The pore formation in the host cell membrane disrupts the normal physiological homeostasis of cells and causes their death. This is known as pyroptosis. The cytokines released stimulate the nerve endings in the interstitium and initiate cough reflex. The cough is dry as the respiratory barrier is still intact. Moving on to the cell or tissue destruction caused by SARS-CoV-2 infection, which happens by three mechanisms. Number one is pyroptosis. Number two is destruction by the NK cells. And the third one is quite interesting. Any cell, normally any cell which is infected with any pathogen releases a cytokine named interferon beta, which alerts the neighboring cells of a possible viral attack, basically sending a signal to the surrounding cells to up their immune response. But COVID-19 is unique as SARS-CoV-2 inhibits the release of this interferon beta. So the cells in the vicinity are not prepared to combat the virus. The ACE2 receptors are present abundantly, not just in the lungs, but also in the kidneys, heart, blood vessels, GIT, and the brain. The lungs are the first target organs of COVID-19, as we all know. But of course, some patients manifest gastrointestinal symptoms like diarrhea first, as some viral particles may happen to escape the gastric acid because of their protective lipoproteinaceous covering around them and enter the intestines, where they bind to the ACE2 receptors and cause infection. Moving on to the ACE2 receptor destruction and the resulting inflammation. We are all aware of the RAS system, wherein renin from the kidneys converts angiotensinogen to angiotensin 1, which is further converted to angiotensin 2 by an enzyme known as angiotensin converting enzyme 1, ACE1, produced in the lungs or the pulmonary vasculature more precisely. The angiotensin 2, when acts on AT1 receptors, causes vasoconstriction and inflammation. These effects of angiotensin 2 are kept in check by ACE2 receptor, which converts angiotensin 2 to angiotensin 1. This is necessary as if in a normal individual, if there is continuous production of angiotensin 2, there would be progressive inflammation and vasoconstriction, which does not happen normally. So it has to be, so the angiotensin 2 has to be degraded to form angiotensin 1 through 7. So basically, there is so the angiotensin 1 through 7 causes vasodilation and has anti-inflammatory effects. So basically, there is a balance maintained between angiotensin 2 and angiotensin 1 through 7. When SARS-CoV-2 in COVID-19, when SARS-CoV-2 binds to the ACE2 receptors, there is competitive inhibition of ACE2, which causes increased levels of angiotensin 2 and decreased levels of angiotensin 1 through 7. Which means that there is more of pro-inflammatory effect and less of anti-inflammatory effect. How angiotensin 2 causes inflammation is when it binds to the AT1 receptors, it leads to the production of reactive oxygen species or ROS, which causes the recruitment of macrophages, which in turn release cytokines. Now, the normal immune response with mild to moderate COVID-19 is as follows. Firstly, there is antigen-independent innate immunity which is activated, and so cells like neutrophils, eosinophils, basophils, NK cells, and mast cells are monocytes, macrophages are recruited. Following this, the lung-resident dendritic cells which are found in the pulmonary capillaries, the alveolar septa, are activated and changed to antigen-presenting cells. These activated APCs or antigen-presenting cells ingest and process the viral antigen and present to the naive THO cells in the lymph nodes with the help of MHC or major histocompatibility complex. Then the naive THO cells differentiate into T helper cells and the cytotoxic or killer T cells. The T helper cells further differentiate into Th1 and Th2 cell types. The Th1 cells release pro-inflammatory cytokines like interferon gamma, TNF alpha, IL-1 beta, IL-12, whereas Th2 cells release anti-inflammatory cytokines like IL-4, 5, 9, 10, and 13. So it seems that there is a balance between the anti and pro-inflammatory cytokine networks. This balanced immune response can limit the progression of COVID-19. But in some patients, there is dysregulated immune response, which can be attributed to high oxidative stress due to excess ROS production, which can be due to high levels of circulating undegraded angiotensin II, which can be either due to increased competitive inhibition of ACE2 receptors or due to decreased expression of ACE2 receptors, as what happens in the elderly. Finally, there is increased ROS production leading to macrophage recruitment, which would in turn release cytokines like interferon gamma, TNF alpha, IL-1, 6, 8, out of which IL-6 is the most important and is being held responsible for the rise in increased levels of acute phase reactants like C-reactive protein or CRP in COVID-19 patients. This overwhelming immune response is what you call the cytokine storm. It is also referred to as the macrophage activation syndrome or MAS, since the activation of macrophages results in massive release of cytokines. Well, by the way, it is also known as, cytokine storm is also known as cytokine release syndrome or CRS. Yeah. Now, if there are high levels of circulating cytokines, they would result in the damage of the vessel wall, the tight junctions in between the endothelial cells, and the basement membrane, causing loss of integrity of the capillary membrane, and thus leading to exudation of plasma and cells like neutrophils and macrophages into the interstitium. Now, when this fluid leakage happens in the lungs, it leads to ARDS. In the lungs, the SARS-CoV-2 damages the cells in both the alveoli, lining the alveoli and capillaries, thus damaging the alveolar capillary membrane or the respiratory barrier by the damaging processes I mentioned before. The alveoli thus get filled with not just fluid, but also with blood, broken tissue, broken inflammatory cells like neutrophils, broken pathogens, etc. This leads to pulmonary edema, which causes productive cough in that patient. At this stage, the alveolar spaces have accumulated some amount of edematous fluid and also forms a hyaline membrane out of the cellular debris and proteins. This incomplete filling of the alveoli and hyaline membrane formation gives the appearance of ground glass opacities or GGOs on HRCT. When this congestion progresses further on, the fluid fills the whole of the alveolar space, giving the picture of consolidation on HRCT. This leads to the serious condition known as acute respiratory distress syndrome or ARDS. Now, as the gaseous exchange is hampered due to the pulmonary edema, the patient suffers from hypoxemia, causing low blood oxygen saturation levels. So due to hypoxia, the tissues switch over their mode of respiration from aerobic to anaerobic. Anaerobic respiration gives a lesser amount of energy or low ATP, and also produces lactic acid on top of that. ATP is necessary for the maintenance of the integrity of various cell organelles and membrane channels. Therefore, low ATP would lead to the damage of the cell membrane and the channels. Thus, an electrolyte imbalance starts occurring, which results in seeping in of water into the damaged cells. This is termed as cell swelling. The cells then start leaking out their contents through the damaged channels, which means that they are now dead and they cannot be rescued. They can be rescued only at the stage of swelling and not when they have leaked out their contents. This rescuing can be done by administering supplemental oxygen, giving ample amount of fluids and electrolytes to correct the BP and restore blood in the damaged area to supply enough oxygen there and to wash out the toxic lactic acid. Now, as we know that there is loss of fluids from the blood vessels into the extravascular compartment due to exudation. And if this happens on a large scale, there would naturally be a fall in blood volume, leading to a drop in BP, which leads to septic shock. but the percentage of COVID-19 patients landing up in septic shock is 19% of hospitalized patients, out of which 3 to 5% land up in critical condition, and unfortunately 3% even die. In the blood vessel endothelium, as I've already mentioned, are the ACE2 receptors, which are the site of binding of the SARS-CoV-2, leading to the release of cytokines via the NLRP3 inflammasome pathway. Also, there is unconverted angiotensin 2 responsible for more inflammation. This leads to endothelial damage that exposes the broken vessel wall to the flowing blood, thus attracting clotting factors like 1 Willebrand factor that acts on this site and causes platelet aggregation and fibrin deposition. And this fibrin is what is responsible for increased or elevated D-dimer levels in COVID-19 patients, thus forming thrombi. The rupture or breakdown of these thrombi results in the production of emboli, which, when travel downstream into smaller blood vessels, would cause the necrosis of the tissue below. This state of hypercoagulability is responsible for the lodging of emboli in various organs. Yeah. So this is a diagrammatic representation that I made to make the systemic pathology simpler by linking some organ systems involved in COVID-19. So COVID-19 pneumonia leads to ARDS, which causes myocardial oxygen supply-demand imbalance, since there is hypoxic damage to the myocardium, plus there is increased workload on the poor myocardium, as there is as the rest of the body has demands high oxygen, which is already in the state of hypoxia. This leads to cardiac failure. In viremia, the SARS-CoV-2 may reach the myocardium, where it binds to the ACE2 receptors and cause pyroptosis and release of cytokines, which would damage the myocardium and cause inflammation, and that is termed as myocarditis, which leads to cardiac failure again. There can be RVF, right ventricular failure, which can also be precipitated by COVID-19 pneumonia. And there can be left ventricular failure too, which naturally results in decreased cardiac output, leading to decreased arterial filling, causing hyperperfusion of organs. And if this hyperperfusion happens in the kidneys, it causes acute kidney injury or AKI. The SARS-CoV-2 in viremia may also reach the kidneys and bind to the ACE2 receptors expressed on the renal tubular cells and the podocytes in the Bowman's capsules. So just like in the myocardium, there is pyroptosis and cytokine release, which causes acute tubular necrosis or ATN. Since the podocytes are damaged, and the Bowman's capsules start losing their filtering capacity and hence leak out proteins and blood cells, causing proteinuria and hematuria, which leads to acute kidney injury again. Since there is endothelial damage by the cytokines, the clotting process is initiated, thereby forming thrombi, which disintegrate to form microemboli, then which may lodge in the cerebral circulation and cause ischemic stroke, or they may lodge in the renal vasculature and cause AKI. They may also lodge in the coronary circulation and cause acute coronary syndrome, which in turn leads to acute cardiac failure. Finally, to conclude, the novel coronavirus binds to the ACE2 receptors in the human body, but the pathogenesis of COVID-19 isn't just about this binding. It's our immune system too, which is responsible for the progression of the disease to an extent of landing up in a critical condition. Thank you all, and especially Dr. Vijay Mohan sir, to have given me the opportunity to be a part of this great show and to be able to share the same platform with the other great, the virtual platform with the other great speakers like you all. Thank you so much. Have a nice day.